We should move on to our next talk. Karthik Gangavarapu, who is from Scripps Research, CViSB here. Karthik has been leading a lot of the genomic analyses in SARS-CoV-2 and other viruses, and also is the lead author together with Al on the IVAR software, which I mentioned earlier today. Karthik.
Yep. I'm Karthik. I'm a PhD student in the Anderson and Sue Labs at Scripps Research, and today I'm going to talk to you some of the work we've been doing on the early transmission of SARS-CoV-2 in the US, with a particular focus on New Orleans in Louisiana. So New Orleans had a first wave of infections that both started early and progressed very rapidly. Here I'm showing you the number of daily COVID-19 cases normalized by population of New Orleans and two other metro areas, New York and Seattle. And as you can see that the New Orleans peak was both early and quite significantly higher compared to other metro areas. I pulled this plot out from Outbreak.info that Laura talked about in the morning. The first case of COVID-19 in New Orleans was actually reported on March 9th. And as with any sort of genomic epidemiology study, the first thing we had to do was, well, get genomes. So with the help of collaborators, we got samples and we sequenced about 152 genomes. I'm showing you the temporal distribution of the genomes below here. And as you can see, the number of genomes closely follows the number of cases over time in New Orleans. Before sort of going into the results, I want to briefly give an overview of the methods. The phylogenetic analysis we primarily relied on machinery implemented in BEAST that Shang very elegantly described in his talk before. I am just going to give you an overview of how to interpret some of the results. In this cartoon here, imagine you have three different genomes. Typically, when you sequence a genome from a sample, you know when the sample was collected. So every genome has a timestamp. You also know the location at which the individual was sampled. So it also comes with a geographic location. If you imagine these three genomes here sampled at three different times, T1, T2, and T3, with the three different colors representing three different geographic locations. As you all very well know, as RNA viruses replicate, they accumulate mutations due to the error-prone replication process, and they accumulate these mutations in a clock-like manner. Using the timestamp on the genomic information, you can actually estimate that clock rate and use that information to then estimate how long it would have taken for different genomes to diverge from each other. As you can see here, these two genomes have an ancestor, hypothetical ancestor that probably arose at time T5, which then arose from another ancestor at time T4. Another term you'll probably see during the presentation is TMRCA, which is time to the most recent common ancestor. The most recent common ancestor of these three genomes is actually this node here, when the TMRCA is T4. Now, in the same way that we estimated the clock rate at which the virus is accumulating mutations, you can also estimate a clock rate at which the virus is sort of changing geographic locations. So you can estimate that this ancestor here was sampled at a location represented by yellow, which arose from another ancestor that is represented by blue. As with any sort of analysis method, the bias in your underlying data has to be accounted for. In particular, if you imagine this to be the true phylogeny, and say in your sampling, you actually missed this genome completely shown in red, then you would sort of wrongly interpret your results that the virus actually moved from location 1 to 3, whereas in fact, that is a direct consequence of the underlying bias in your data set. Given that early on in the outbreak, we were definitely massively undersampled, this is a bias that actually exists in our data set. In order to account for this bias, we actually present a secondary line of evidence using travel risk to support any hypothesis. The number of reported cases of COVID-19 is highly biased by delays in reporting and testing early on. So we assume that the number of deaths reported due to COVID-19 is actually far less biased. From previous literature, we have a good idea of how long it takes an individual to go from infection to death and the infection fatality rate. So using those two estimates, we actually estimate the number of daily infections of COVID-19 over time. Now, in order to actually estimate travel risk, what you need is for any given location, you want to estimate the number of infectious travelers at any given point of time. Again, going back to literature, we have a good sense of how long an individual stays infectious, and we assume that an individual starts becoming infectious a day before the onset of symptoms. So using these two distributions combined with the daily infections, we have a good sense of the number of infectious individuals in any given location. We combine that with travel data to then estimate the risk of importation of a virus from one location to another. Okay, so we took the 152 genomes that we had from New Orleans, combined them with a representative set of publicly available genomes. And the first thing we wanted to look at was whether the introduction into Louisiana was domestic or international. In this time-resolved phylogeny here, you see that the sequences from Louisiana clustered primarily with sequences from within the US, showing that the introduction into Louisiana was most probably domestic. This is also supported by air travel data, where you see most of the travel coming into New Orleans has a domestic origin and not an international one. When you actually look at the genomes in New Orleans, you see that the genomic diversity of these sequences is actually significantly lower. I'm showing you the different lineages that were found in every different location. You see that the heterogeneity and diversity is actually very comparable to the diversity that you see in outbreaks that happened on cruise ships, showing that there was probably a very limited number of introductions of the virus into New Orleans, and these introductions were probably also very homogeneous in terms of genomic diversity. And in fact, when we look at the phylogeny, we see that there was a single major introduction of the virus into New Orleans, which then caused the majority of the cases in the first wave of infections. In this phylogeny here, this gradient here represents the uncertainty in the tree topology. Keep in mind that most of these genomes have been sampled within a period of a month or a month and a half. SARS-CoV-2 typically accumulates one mutation every 2 weeks. So there's actually very low genomic signal here to actually estimate the clock rate. But we were able to get a well-supported estimate for a single introduction into New Orleans. When we look at when the virus most probably jumped into New Orleans, we think it probably happened sometime in mid-February with a median time of February 11th. The next thing we looked at in the genomic data was actually the source of this introduction. And here we see that we think from the genomic data, we estimate that the epidemic in New Orleans was most likely connected to Texas. I say connected to here, alluding back to the bias in our underlying data set, where we don't know if the virus sort of directly jumped into Texas, or there was an unknown intermediate location where we didn't have genomic sampling from. However, when we look at the travel risk, it tells a very similar story. So we estimate that the introduction happened on February 11th, which is week 7. And when you actually look at the travel risk, the risk of importing the virus from a certain location, Texas ranks among the top 2 places as posing a risk to New Orleans. This is in line with what we see in genomic data showing that the epidemic in New Orleans is most likely from Texas. Now, I mentioned early on that New Orleans had the first wave was early and progressed very rapidly. When we actually model the number of infections from daily deaths, we see the same trend hold. Here I'm showing you I'm comparing the infection curve in New Orleans to other metro areas in the US, and you see that compared to all of them, it started earlier and progressed more rapidly. The one event during this time period that sort of sets New Orleans apart is Mardi Gras. And in fact, when we look at the first introduction, the introduction of the virus into New Orleans, it happened February 11th with 80% probability that it actually happened before Mardi Gras. As you all very well know, Mardi Gras attracts over 1 million visitors into New Orleans every year. And this signal is also seen in the travel data, where we see a spike in travel from other states coming into New Orleans during the week of Mardi Gras. Now, we actually wanted to quantify the number of cases, the number of infections that were probably caused on the day of Mardi Gras itself. In order to do this, we went back to the modeled infections from daily deaths. And we see that when you look at the cumulative number of infections, that a median of 792 cases are required to recapitulate this entire infection curve. Now, this model is completely unaware of a large event like Mardi Gras actually happening. So we took this cumulative number from here, and then we know that there was a single introduction of the virus into New Orleans. So then we simulated the number of cases over 4 generations, starting from February 11th until the day of Mardi Gras. In this simulation, what we do is we assume that the number of secondary cases caused by any primary case can be described by a negative binomial distribution, which is described by an R0 and K being the overdispersion parameter that accounts for the variation in transmission of SARS-CoV-2. So we simulate this 100,000 times, and then we end up with a cumulative number of infections from the simulated data about the number of cases that probably happened until the day of Mardi Gras. Since both these models are sort of unaware of any event like Mardi Gras happening, we account for the difference between the estimates of these two from the cumulative infections from the daily infections and the simulated data to actually show that, to actually quantify the number of cases that happened in Mardi Gras. And we see that there's a 94% probability that at least 100 different infections occurred on Mardi Gras itself, showing that Mardi Gras was most definitely a super spreading event and played a critical role in accelerating the first wave of infections in New Orleans. The next thing we looked at was how much risk New Orleans actually posed to other locations within the US. Here from the genomic data, we see that New Orleans probably caused localized epidemics within Louisiana and also in the neighboring states of Texas and Florida. When we look at travel risk, it tells a similar story. Here I'm showing you the risk of importing the virus from New Orleans into all the different states. You see that for most states, the risk is actually well below 40%. The one state that stands out is actually Mississippi, which there's almost a 60% of importing the virus from New Orleans, showing that New Orleans is probably one of the major sources of the epidemic in Mississippi. Now, as we were sort of wrapping up this story, that was the time when the first cases of the B117 lineage were actually detected in the US. So the B117 lineage, as you all very well know, was first detected in the UK and has been shown to be more transmissible in UK and Denmark. I'm going to briefly talk about the results of the study there that we did along with collaborators from Helix, but I'd be happy to talk about it more after the talk. So we looked at the testing data from Helix and we fit a logistic growth curve here to actually predict the number of the proportion of B117 cases over time. We see that the lineage is 35 to 45% more transmissible compared to previously circulating lineages in the US, and the cases caused by this lineage are doubling every 10 days. We estimate that if the lineage continues to spread at this rate, that more than 50% of the new COVID-19 cases would be caused by the B117 lineage in the US sometime around mid to late March. Along with testing data, we also looked at the genomic data. We had about 212 genomes of the B117 lineage in the US. And when we actually look at the phylogeny, you see that there were at least eight independent introductions of that lineage into the US from outside the US, with 2 major introductions coming into California and Florida. When we look at the timing of these introductions, we see that most of these introductions actually happened between late November to early January. And as more and more data comes in, we'll be able to fill out the number of introductions and establish a more accurate story as to how the lineage is actually spreading. With that, I want to thank our collaborators, without whom both these studies would not have been possible. The Gary Lab, Jeremy Campbell, Juan Cooper, Suzanne and Rebecca from Bioinfo Experts for giving us samples and viral genomes for the New Orleans study. the Ghana lab for their help with modeling the travel risk, Gides and Laura for their help putting up crafting the actual story, Mark and Philippe, who played a critical role in actually helping us set up these analysis and interpreting the results, Katie and Mark, who lead the sequencing of SARS-CoV-2 in the Anderson lab, folks from Helix, who helped us with the B117 study, and finally my professors Andrew and Christian. And that I'm done. I'd be happy to take any questions.
Thank you, Karthik. That was great.